Hello everyone, so again, this is going to be a continuation of that Captain's Log study journal review about some information I learned about in one of my classes. Hopefully I could pass on this information to all of you. It's on my microbiology class, so we're going to be talking about the different types of microbes that we'll be studying throughout the year. So, in the microbial world, we have a few main characters here. So we have the living organisms, such as the bacteria, Archaea, as well as the Eukarya. Eukarya breaking down into the protists, which break down into algae, as well as protists themselves, so protozoa. Then we also have our fungi and some helminths. Alright, so on this side, bacteria, those are the single-celled prokaryotes. They're unicellular. Archaea are just basically bacteria that live in more extreme conditions. They're able to take a lot of punches. They're able to roll with things such as high saline content, as well as high temperatures, even low temperatures too, so they're a very interesting group. Eukarya, these are living things that have membrane-bound organelles and compartments. We have the protists that are make up the protozoa and the algae. And then we also have our fungi in the form of yeast. And there's also some other microscopic species of fungi. Just yeast is one of the more simpler ones. And then helminths are worms. So these are parasites. And you might be wondering, oh, but I could see a worm using just my bare eye, usually. Well, worms, is particularly in the ones that are parasitic, they do have microscopic eggs as well as larvae. So that's why they're included in our microbial world. Other members of this pantheon include things such as the acellular, infectious agents. These include viruses, Viroids, and Prions. These are non-living. They're not alive. These things are as dead as a piece of rock or something like that. They're literally not alive. They do not use or give off waste. They do not need energy. They don't take in energy. They do not reproduce on their own. So these are non-living. Alright, so let's start with the domains. Let's start with, again, our good old friends. They've probably have met them ever since you've been taking science classes. We have the prokaryotes and the eukaryotes. Alright, so prokaryotes, no membrane bound nucleus. While over here we have organelles. And usually there's also a level of complexity with these organisms. Alright, so if we go back here to our microbial world, our little treasure hunt map of all the different microbes that we might be studying. Prokaryotes include bacteria and archaea, and eukaryotes are basically everything else. So eukarya, all of these things, fungi. helminths, protists, algae, protozoa. Acellular infectious agents, however, aren't alive again, so they do not fit in either category. Speaking about microbes, we could also talk about the sizes of things. So, the unaided human eye. So let's draw just regular eye, kind of like how they teach you in like an intro to arts class, where you have, you know, an almond shape, and then could, I guess, put some eyelashes. This looks really strange, I know, but we're trying our best there. And then, you know, there's the lines that go from the pupil. All right, so the human eye, we could actually see anything from 1 millimeter, a little bit less than 1 millimeter, so I'm going to actually do that, or more. So we could see everything big up here, so like a person, and then sometimes a large bacterium. You could kind of see that sometimes with, there is a species of bacteria that you could see with the unaided human eye. All right, afterwards we have our light microscope. So I'm just going to put light, and that will go from 100 micrometers to about 1. These are approximate again, so here we will be seeing eukaryote cells, as well as most bacteria, some of the organelles. I'm going to just put most back, and then nucleus as one of the main organelles we will be seeing. Made a spelling error. No, I did not. All right. Now we go into the electron microscope. This is an even more powerful tool, so that way we could see even smaller objects, up to 1 nanometer. So this again goes all the way to 1 nanometer. All right, so here we will be seeing viruses, so I'm just going to draw like COVID-19 to exemplify a virus. They're very tiny particles. We also have ribosomes, so ribosome usually has 2 subunits like that, so there's a ribosome. And you could even find some larger molecules, such as some larger lipids, so we could draw like a random molecule here. And then beyond that 1 nanometer, when we get to even smaller increments, like 1/10 of a nanometer, that's when we start seeing individual atoms. Alrighty. So now let's go into each of these different types of organisms, as well as the non-living particles in microbiology. So let's start off with our friends, sometimes our enemies, the bacteria. So again, prokaryotic. And this means they don't have their DNA in a nucleus. Instead, it's in something called a nucleoid. They don't have any organelles that are bound by membranes. So, you know, bacteria sometimes have different shapes. There's something here, this is like a berry-shaped bacteria called coccus, or cocci if there's more than one, so we could actually just call it cocci. This is kind of like a rod-shaped bacteria. It's referred to as bacillus, and so forth and so forth. We also have a cell wall, but this is unique in bacteria, where you have either a lot of peptidoglycan or no peptidoglycan. And peptidoglycan, it's this molecule that kind of has a lot of sugars in it, kind of has some protein elements to it, and they use this for their cell wall. Some species of bacteria will have peptidoglycan and a thick layer of it on their cell wall, and these are called gram-positive. However, if the bacterium does not have that thick layer of peptidoglycan, then it is known as gram-negative. Bacteria do multiply by binary fission, which is when one bacteria here, they just split kind of like so until there's two bacteria. So kind of like mitosis. And they move by using something called a flagella, which is this little flag or little hair-like structure, and then that just propels them, just like that. Zooming everywhere, look at that. Okay, let's go into our next prokaryote, the Archaea. This is the domain Archaea. And these are very similar in bacteria. Because of their domain is in the classification of being prokaryotic. They also have the similar shape and the size and how they reproduce. Remember, that's the fission. They're reproducing by fission. Flagellate movement, as well as cell walls. However, the cell walls, I should say, not the cells themselves, but the cell walls don't have any they don't have any peptidoglycan in them. So that's a difference. Alright, so they also have ribosomal RNA, or ribosomal RNA. It's also known as ribosomal rRNA. These are their genetic sequences. And they are extremophiles. So, what is an extremophile? Well, the root of the word, extreme, extremo, and then phile, meaning loving. So these are very, very attuned to very high salt concentrations. So we could draw like a salt shaker maybe. Let's put salt there. So high salt. They also like high temperature. So it's going to probably put like a bonfire to show that. They also like high temperature. So they live in these extremely brazen as well as difficult living conditions. And they're able to thrive in these very, very harsh conditions that usually nothing else would live in. Alright, so now we are in the domain Eukarya. Welcome to the domain Eukarya. Alright, so in here, the largest thing is that we have membrane-bound organelles. So, such as the nucleus, all of those things. There's a high level of complexity. And the members in microbiome include fungi, are worms, so helminths, as well as algae and protozoa. Alright, let's get started here on fungi. Okay, so these could be single-celled yeast, a very simple, easy-to-manage life form used for the production of course, beer, bread, kind of look like little spheres if you see them under the microscope. They're a model organism because they're easy to maintain. They reproduce in a short amount of time, so you could run some tests on them if you're looking at something genetic-wise. You could also look at multicellular fungi, such as mushrooms or molds. And then fungi are decomposers, so they take in dead matter or decaying matter and recycle it, and they are usually based on land. Alright, next up in domain Eukarya we have algae. All that stuff in the water, so if there's like a lake and you see patches of all these plant-like specks and whatnot, that isn't a plant, that's actually algae. Alright. They could be single-celled or multi. They're photosynthetic. This is a very unique property of algae. Compared to some of the other microbes, yes, there are photosynthetic bacteria, but for the most part, algae, they're going to be photosynthetic. They contain the chloroplasts that plants do have as well. And alongside chloroplasts, you know, they'll have their phylls. So chlorophyll, all the other types of pigments. They are usually aquatic. As we were talking about earlier, they're in the ponds everywhere you'll go to a lake, you'll find them. They have cell walls that are really rigid. And they have flagella. That's a big indicator that you're looking at algae, because some of the prokaryotes will actually have different flagella than looking at an algae flagella. So you could possibly tell them apart if you're seeing something that looks like it has a chloroplast or photosynthetic capabilities. If you just see the algae flagella, then might be something that you could use to differentiate. Okay, now protozoa. These are fun. Protozoa, basically, these are like the junk drawer of living things. They usually are very weird in like how they would fit into like the tree of life. So that's why they get the term junk drawer. However, they're very complex. That's why they are given the term first animal, proto, and then zoa, first animals. Even though they probably aren't the first living things, mind you. They're given this because it's kind of like a catch-all. There's a lot of interesting, unique species in the protozoa. Mostly, they could be complex. They're usually larger than prokaryotes. But they're all single-celled. And they are usually predatory, so they are predators. And they also ingest other organic compounds too. So they're consumers. And they don't have a cell wall, so no wall. We don't need a wall, say the protozoa, alright? And most of them are motile. You have things in here such as amoeba that have these weird, like, pseudopod or fake feet projections that they use to move around. You also have things such as a paramecium or plasmodium that kind of look like that. And then there's a ton of them. They're just single-celled. They're very interesting, very unique. You could see a ton of organelles if you look at them under the microscope. You could see a ton of things going on in there. Very interesting. Right, we also have the parasitic worms, also known as the helminths. And these are microscopic at the egg and larva stage. These are usually going to be seen without magnification at their adult stages, but they do include roundworms, tapeworms, and flatworms, aka the fluke. Alrighty, now we are in the interesting part. We're looking at acellular, infectious agents. And I'm going to switch to a different color since we're in a different section now. Alright, the largest one here, viruses. Non-living, again, this is a trait for all these acellular infectious agents, non-living. Okay, so basically a virus is at the shortest, most basic level of kind of like comprehending what a virus is. It's just a bunch of nucleic acid. It could be DNA or RNA, either one, in a protein coat. And then you have a variety of shapes. You know, you have those ones that look like space invaders, like this. And then you also have, you know, your coronavirus. As of making this video, there is a new variant going around, so they had some talks about kind of warning everyone about that, just in case. And those are two shapes that you usually see viruses in. They infect living cells, so they kind of do this thing called hijacking. They term the cells that are hijacked, they call them hosts, so they hijack host cells. Just kind of put two S's there at the moment. And they do this to multiply, and they also use this to take up nutrients. And this helps them build more viruses. And the interesting thing about any of these viral particles, outside of the host, they are actually dead. They can't do anything on their own. That's why they're obligate intracellular parasites. And all forms of life have viruses. We all have them. So from a skunk to a mushroom to a blade of grass to a human, we all have viruses. Alright, now let's go to something a little bit more simpler than a virus. This is called a viroid. Basically, you have a virus, and then they only have a piece of RNA, that's all. Saying that, they do not have a protein coat, so no coat. They do the same thing as a virus, but specifically they cause plant disease. So here's our long infectious piece of RNA, and there we go. So this is going to probably be a viroid. We just need RNA. There we go. Much more of a simpler infectious agent. Alright. Prions. I do not know how you actually pronounce it, so apologies about that. Another YouTuber actually did an amazing video on prions, and I recommend you checking that one out. In the presentation, prions are actually misfolded infectious proteins. Yes, so they are proteins. They aren't a living thing. Sorry about my spelling error there. They are not living. They are just misfolded proteins that are within the brain, and when they make contact, let's say that this is a prion, right? Let's say that this is a perfect folded protein. So this protein is good. This one is the prion because it's not folded correctly. And this is taking place in a person's brain. So I'll just put brain in big red letters. Okay, so the prion on the left, this one, let me select it. What happens, it will come into contact with this good folded protein in the brain, right? And then what will happen is it will cause that one to also become misfolded like so. So now this is going to be called a fibril. Now the once well-folded protein has become misfolded because it came into contact with the prion. It's now called a fibril. Fibril. I do not know how to pronounce that. Oh well, I must have screwed it up. Anywho, the effect of the fibril forming, this causes a loss of function. And look where this all takes place. It takes place in the brain. This is why prions are very dangerous, because you get all these degenerative diseases that happen once you shut down the seat of operations, where all the operations are being managed and controlled, aka your brain. The whole system just starts shutting down. This is where we get neurodegenerative diseases in humans and in animals. So prions are very dangerous, as well as viruses. Of course, we went through a pandemic. It's all about being informed, staying informed, and making sure you're taking the best steps to protect yourself and get information on the microbial world. All right, so thank you so much for tuning in. Please do something nice for someone.